Thank you. The mic is on, I can tell. Is it too loud? It's okay? So it's a pleasure for me to be here. This is a particular challenge. I'm used to speaking, but not to such a diverse group. So I've prepared. I feel confident, but if I don't get it right, you can interrupt me and ask questions. In fact, I encourage you to do that, and I'll give you some opportunities to ask questions as we go. So I'm going to talk about some of what biomedical engineers do, and I'm going to focus on things that we've done in my research lab here. And obviously it's a big field, so I'll only be talking about a small part of it. And I've given myself a title which I couldn't possibly live up to, Medicines of the Future. That's a big topic. So I'll say a little bit about what I think medicines of the future will look like. Some of them are actually medicines that are now being used today, but we're going to be delivering them in different ways, and they're going to be useful for treating diseases that we have difficulty treating now. So that's my topic. And as Professor Zilm mentioned, I grew up in Iowa, and I know there's a couple of people in the room, but hopefully most of you know where Iowa is. It's going to start off as a geography lesson, if not. So Iowa is here. I grew up here. And for some reason, which I can't explain now, I was always interested in math and chemistry and sort of science in general, but math and chemistry in particular. And it didn't come from my family. My grandparents were all farmers. This is what Iowa looks like, not everywhere, but in many places. This is my grandfather's farm, which was actually his grandfather's farm in the very southern tier of counties, Ringgold County, Iowa. And a lot of science happens in places like this. So maybe that's where I got exposed to interest in natural phenomenon and how to make things work. But it's a long way from that to here. And I'm not going to say very much about the story. I'd be happy to answer questions about how I got here if you like at the end. But if you would have told me when I was a middle school student or a high school student that I'd be a scientist and that I'd be teaching at a place like Yale, you might as well have told me I was going to win the lottery, and I just didn't think it would happen. It wasn't something imaginable. So I'm happy to be here talking to you about what we do and feel very fortunate to be in this position. So I encourage all of you who haven't started your careers yet to be open to whatever happens. You don't know. Remarkable things can happen to you, particularly if you pay attention to math and science. Just as further background, these are the courses that I teach at Yale to sort of give you an idea of what I've come to be an expert in. I teach an introductory course in biomedical engineering, and I saw Durga here earlier. A Yale student who took the course. There she is. She was one of the students giving you demonstrations. So this is to introduce people to the field, and I'm going to give you a little introduction to it today. I teach physiological systems, and this might seem strange to you that an engineer is teaching human physiology to future doctors. But really, we understand physiology so well now. We understand how all of our organs are built that one can understand them in terms of the mechanisms by which they work. And it's really engineers who can sort of synthesize the information that biologists have and understand how to present it, I think. So that's how I ended up there. I teach a course in drug delivery and design, which is the topic I'll be talking about. And I'm very interested in how to do things in the brain, and that will become clear as we go. So this is sort of the outline for what I want to talk about. New ways for giving medicines, new ways for giving drugs to make them more effective, and in particular to make them safer. One of the biggest problems we have when we develop medicines to treat diseases, and this is particularly true for diseases of the brain, one of the biggest problems we have is that they cause side effects in other places that often are worse than the disease you're trying to treat. And so how could you take medicines that you know work well and give them to people so that they don't cause these side effects? So I'm going to talk about that in the context of treating cancer, which you all know is a terrible set of diseases that we still don't understand completely and we still don't know how to treat. And then I'm going to talk about something that I think is even more forward-looking, the idea that one could use molecules to cause tissues to regenerate in people, that one could put chemical systems together in ways that allow you to build new tissue in tissues like in the brain or in the heart that are failing. Okay, so this is the brain. Who knows which is the front? Where's the forehead? Here. Do you know which side? This one or this one? Yeah, this is the front of the. I should be pointing to this one, right? This is the big one, but that's the one I see best. This is the front of the brain, so this is the forehead right behind here. This is the back of the head right here, and this is the part that we really value. This is the cerebral cortex that you can see on the top, and this is really the part of the brain that makes your whole body work. There's parts of the brain that allow you to control your arms and your legs and your muscles. There's parts of the brain, particularly in the front, that help you to decide what to do today and what to do tomorrow, that allow you to think. And we know that there's lots of things that we use our brains for. We're all using them now. We use our brains to learn about the world. This is a group of public school teachers who came into my laboratory a couple of years ago to learn some about the things I'm going to tell you today. And you can see that they're all focused on learning from Chike, who's a medical student, who's showing them his particular experiment down here. And people have all different ways of learning. You can see some are really intensely learning and some are sort of learning on the periphery. But we're all using our brains to learn about the world in different ways. We use our brains to interact with people and materials. This is my son, Alex, and my niece, Ruby. And we use our brains to help each other by activating muscles to hold Ruby off the ground here, for example. And we use our brains to predict the future. And that's what my son, Zach, is doing, I think, in this picture here. So when we have diseases of the brain, there's a lot of interest in trying to cure them or to restore function. And biomedical engineers have been very heavily involved in this. One way they do it is by designing systems that let you look inside the body without using a knife or a scalpel to open it up. In the old days, if you wanted to see what was happening inside the brain, the only way to do that was to open up the brain and look inside. And obviously that's a challenge. But this is a picture from a technique called magnetic resonance imaging, where you can see in quite a lot of detail that brain that I showed you a cartoon of before. Here's the forehead, here's the brain, here's the back of the brain, the cerebellum, the spinal cord, and you can see those regions of the cerebral cortex that I talked about earlier. Sometimes special regions of the brain fail. In Parkinson's disease, there's a set of cells that are deep inside your brain that stop functioning in the normal way, and you're not able to control muscle movements in the way that you did before. So patients that have Parkinson's disease often have tremors. They can't control their motor movements. They have trouble initiating movements. And biomedical engineers, not completely understanding how this worked, designed systems that are little electronic patches or little electronic devices that are implanted inside the body. And this is sending signals, which are carried in a wire up into the brain. And these little electrodes are stimulating the brain. And they discovered that if you stimulated the brain in the right fashion, you could allow patients that have Parkinson's disease whose brains aren't controlling motion in the right way to control movements better. This is called deep brain stimulation, and now there are many millions of people who have these devices who are able to do the kinds of things I was just talking about with the help of an electronic mechanical device. And surgeons use tools of biomedical engineering all the time to try to understand better how to treat patients. This is an electrical device, a large grid of electrodes which placed on the brain of a patient that has epilepsy, allowing the physicians to find where in the brain, in a patient with epilepsy, these unusual electrical signals that are creating seizures occur. And then once they find where they occur, they can treat disease. So there's lots of examples of engineers, people who build things, devices you can make images with, devices that you can implant, devices that can record how they're helping doctors to improve treatment of disease. This young gentleman has a problem, what is it? He has a headache. Yeah, and we all know when somebody has a headache because bolts of lightning shoot out of their head. What do you do if you have a headache? You take some kind of medicine, right? And how do you get it into your brain? That's where the problem is, right? You've got an ache in your head, and that ache, that pain is originating in your brain. But how does the medicine get there? So I heard something. Through the blood. Through the blood. And how did it get into the blood? You're right on. So you swallowed the pill, and just like, you know?
You swallow the pill and then it digests it, and when you digest it, it goes into your bloodstream.
Yeah, you swallow the pill, it goes into your stomach and your digestive system. Your digestive system is specialized in breaking things down into their chemical components. And then those smaller pieces, like the medicine, get absorbed into your bloodstream, and the medicine goes all over your body, and some of it goes into the brain. But it goes everywhere, right? The medicine goes everywhere. And what you hope is that some fraction of that medicine gets into your brain and you get rid of your headache. But you know there's also medicine going to your liver and your kidneys and your skin and your intestines. It's going everywhere, right? And why can we treat headaches with drugs like Advil or aspirin? Because the Advil or the aspirin molecules don't do anything or don't do very much in the rest of our body, right? They treat the headache and they don't cause any bad effects. But sometimes they do, right? If you take too much aspirin, you can hurt your hearing. You'll start hearing ringing in your ears, and if you keep taking more aspirin, you might even start to lose your hearing. If you take too much Advil, you can damage your liver, right? And so you have to be very careful with how you take medicine. And we're wasting a lot of medicine, right? Because what you really want to do is focus it here, and the rest of our body that's getting this medicine isn't really able to use it. So it's not a problem with headaches, right? Because we have good drugs that you can take that are really safe. It's really hard to damage your liver, so you don't need to worry about that. That is a potential problem, but most of us won't encounter it. Right? Because the drugs that work don't cause damage in other places. Right? And the way that drugs get there is because they go into the blood, and the brain, if I looked inside the brain, is filled with blood vessels. Right? This is a brain where all the brain is removed except for the blood vessels, which are filled with red plastic dye. And you can see that the brain really is a ball, like a tumbleweed of blood vessels. And we need to have all this blood and all these blood vessels in our brain because our brains are using chemicals that are in the blood in order to do all these things that we like for our brains to do. The brain is really special, though, and one reason it is because the blood vessels in our brain are very special. That they don't allow just any chemical to go from the blood into the brain, right? All this blood is moving through your brain all the time. And the molecules are contained within blood vessels or tubes, which are little highways that carry it all over the brain. And most of the drugs stay inside the tubes. Most molecules stay inside the tubes. Only a few drug molecules are able to leak out of the blood capillaries and reach the real working cells of the brain, the neurons. Only a small set of compounds are able to do this. Aspirin, Tylenol, Advil, ibuprofen, that's one kind of molecule. Caffeine is another kind of a molecule that if you drink it, it gets into your blood, and it gets very readily out of the blood vessels and into your brain. And many of us take advantage of that every morning when we drink coffee. You feel a little different after you drink coffee than you did before, and that's because the caffeine molecules have gotten out of the blood vessels and into the brain. Most drug molecules don't. Yeah. So alcohol, I wasn't going to mention it, but alcohol is one of the best alcohol. Do you know what the molecule that makes alcohol work is? It's ethanol, which is a special kind of alcohol. Ethanol is able to move very rapidly from the blood into the brain, and that's why alcohol has effects on the brain. If you ingest alcohol, you start to feel different, you start to behave differently, and it has that effect very potently because it gets out of the brain. Now, how does alcohol have that effect? Nobody really knows what it is that alcohol does when it gets into the brain. I think that was the question that you're asking. It does lots of things, most of them harmful. It has this effect on mood and behavior, and particularly if it's used chronically, it can have very bad effects on the brain. So I didn't answer your question perfectly, and I'm sorry, but it's a really good question and a hard one to answer. So most drugs don't go through as easily as alcohol does. And so the problem with that is that, let's say that there's a very serious disease somewhere in the brain here, like God forbid, a tumor in the brain. What you'd like to do is give drugs, and you'd like those drugs to accumulate in the brain, but most drug molecules don't. They circulate in the body, and very few of them get out into the brain where the tumor cells are, and so they can't kill the tumor. If you know unfortunate people who've had tumors in the brain like I do, you'll know that they do get chemotherapy drugs, right? And the problem with giving them chemotherapy drugs is that they're given by pills or they're given by injections, and they circulate in the blood the way you described. And a few of those molecules get in the brain, but most of them go somewhere else. And so what happens to patients that are taking chemotherapy drugs? They get very bad side effects, right? What kind of side effects do they get? Their hair falls out, and their hair falls out because the drug is going to the skin where it shouldn't be, and it's killing cells that are responsible for making hair in the follicles of the skin. They often get very bad intestinal systems because it hurts the lining of their stomach, which is also always renewing itself. Right? So, and it hurts the bone marrow, so they get diseases of the blood. So years ago, we thought maybe there's a different way to treat diseases like brain tumors, which are life-threatening and very serious and difficult to treat with all the ordinary ways that we give medicines. And so we developed these little wafers. And you can see it here. It's about the size of a dime. This wafer is about the size of a dime, and it's made of a polymer. And you all know what polymers are now because you visited Senya outside. And Senya showed you how to make a polymer called nylon. This is my son, Zach, making nylon in the same way that you did on the bench outside. And nylon is a polymer. It's a very high molecular weight synthetic chemical that can be formed by chemical reactions in a beaker like you did outside. And it has the property that you can make it into a material. And in this case, Zach, like you did with Senya, is made of fiber of nylon. Now, you can make amazing things out of nylon and other polymers, as you know, because it has really interesting properties as a material. In fact, you could mold nylon into a device like this. And you could make a very nice coin or a wafer out of nylon that would be hard, it would be very difficult to break, and you could make things like that from it. Of course, you can make clothing from nylon, and so you can weave the threads like this into fabrics. Now, we knew that you could make polymers like this, but we wanted to make polymers that were special in another way, that when you put them in the body, they'll slowly dissolve. If I made a wafer out of nylon and implanted that nylon into the body, it would stay there, wouldn't change at all forever, basically. And that's not necessarily what we wanted to do. We wanted instead to make a wafer that would have good mechanical properties, so you could pick it up with forceps and you could implant it into tissues, but that it would slowly dissolve in water. Right? That's not a property you want in nylon because you don't want your nylon jacket to dissolve when you're out in the rain. Right? But we did want that to happen in these wafers. And in fact, we wanted them to dissolve in a very controlled way. So when you implant this wafer into a tissue, like into the brain, it would slowly, slowly dissolve. And the nylon, if you looked at this nylon, it's hard to tell in a fiber, but it's relatively transparent. And if you dried it out completely, it would look quite transparent. You can see that this special polymer we made here is white, not transparent. And it's white because we synthesized it around a bunch of drug molecules, and the drug molecules are white. And so this wafer is filled with drugs. Right? So it's a plastic or polymer wafer that will slowly degrade when you put it in water, or you put it in the body, which is largely water, and it's filled with drugs. And so what happens if you drop this into a test tube full of water? What the graph shows here is the drug releasing from the wafer into the water over time, over a period of a couple of weeks. Right? All we did was drop this wafer, which was loaded with drugs, into a test tube full of water, and then we sampled the water every once in a while and asked how many drug molecules have come out of the wafer and into the water. And what you see is molecules, the drug molecules are slowly, slowly, slowly released from this wafer over a period of about 2 weeks. And then over the next month, the wafer itself dissolves and disappears. So you're left with nothing solid any longer. So what could you use this for in medicine? I've already given you hints about this, right? If you could make a wafer like this that slowly released drugs and disappeared, maybe you could use that to treat diseases. Maybe you could use it to treat serious diseases like brain tumors. So here's one of those magnetic resonance imaging pictures of the brain. I'm showing it from a different angle this time. This is like a cut this way. So you're looking at the brain from the top. And you can see inside here, there's a region that doesn't look like it should, and this is a brain tumor. So what do surgeons do once they've identified where a tumor is? They'll try to surgically remove it. Right? So do a surgery to remove the tumor. Why does this work? Because cancer is formed, tumors are formed from clusters of cells that are growing out of control. Right? Cells are dividing, producing more cells, more cells. And if you could remove the cells that are causing the cancer, maybe you can get rid of the disease. And so surgeons have been doing this for many, many years. And it helps, but it doesn't help enough in that most patients who have the most serious form of brain tumors, if you take out the tumor, it will, after some period of time, reappear. So most patients who have the most serious forms of brain tumors don't live for a year or 2 years. They die within a few months, even after you remove the tumor. So we thought, what if we gave these wafers to surgeons as a tool? They're going to remove the tumor, but when they remove the tumor, they could put back into the space where the tumor was. They could put these wafers into the tumor. And this is a picture from the operating room of my friend Henry Brem, who's the chair of neurosurgery at Johns Hopkins. And he really pioneered this technique of taking the wafers that we made in the laboratory and placing them into the place where the tumor used to be. And what's the advantage of that? Now, you don't have most of the tumor cells anymore, and the patient leaves the operating room with these devices that are slowly releasing drugs. The drugs are in the brain. They're not in the rest of the body. So the drug is right where the tumor is. Hopefully they'll kill the tumor cells more effectively. And even more importantly, they won't go to the rest of the body. These molecules are now trapped in the brain, and so they won't go to the skin and cause hair loss. They won't go to the bone marrow and cause anemia. They won't go to the intestine and cause intestinal disease. This makes sense as an approach. So how would you test if that worked? We tested it in animals. These are rats that have tumors. And what these curves show you is these are animals surviving as a function of time. So animals who have brain tumors, this kind of brain tumor, they don't live for very long. From when their tumor starts, 2 weeks, most of the animals are not surviving anymore. Right? But if you give them one of these treatments by implanting a polymer that contains a drug called BCNU in the brain, they live much longer. On average, they live 6 to 7 weeks. And some of these animals are cured of their tumors. You never see the tumor coming back. And you have to test it in animals before you test it in people because you want to make sure that you've done the engineering right so that the thing that you're implanting in people is completely safe. And that's why we did all these studies. But eventually it was tested in human patients that have tumors. And what do you see here? These are patients who go to the operating room to have their tumors removed. And when they get the tumor removed, they either get a polymer that didn't contain any drug, or they get a polymer wafer that contained the drug BCNU. And the patients that were lucky enough to get the polymer that had the drug in it lived longer, and some of them lived much longer than you would expect based on this very serious disease. So I realize I was explaining a lot of things here with graphs, but what do you think of this result? On the one hand, we were very happy because this turned out to be, this was approved by the FDA in 1996 and has become available for patients all over the world. And that's because we shifted the survival curve so that patients are living longer, but they're not living that much longer, right? So we made something new and it worked and it helped people, but we haven't totally addressed the problem, right? Because patients still don't live very long with this serious disease. So I was very proud of this. It was the first new treatment for brain tumors in 25 years that had been approved by the FDA. It was an advance in brain tumor treatment, but it's not where we would like to be. We would like this blue curve to be way out here, right? So we thought a lot about why it worked and why it didn't work. And so this is a question I have for you. If you put a drug-releasing implant like this in the brain, what do you think happens to the drug molecules? We learned about what happens to drug molecules when they go into the blood. They circulate, and then they go into tissues all over the body. But what happens when you put them into this, in this special way, into the brain? And so here's one answer. Do drug molecules accumulate to high concentrations in brain tissue? Well, that's what we hoped would happen, that the drug molecules would accumulate to high concentrations. Do they spread from the implant to cover the entire brain? That makes sense, right? The implant's in there for a long time, and so drug molecules should go all over the brain, and they should cover the brain. So we ought to be able to kill tumor cells that are anywhere inside the brain in this approach. Do they move out of the brain and cause toxicity in the bone marrow? No, because they're in the brain, so they can't get out very easily, and they never make it to the bone marrow. So it turns out that the answer to this is two of the above. Two of these things are true and one isn't. Which one isn't true? C. No, C turns out to be true. C turns out to be true. When you put the drug into the brain in this special way, you get almost no side effects outside of the brain. So we've almost completely eliminated the normal kinds of side effects that you have with chemotherapy. So C is true. Of A and B, which one isn't true? A. No, it turns out that one's true. That they do accumulate to high concentrations, and that's why it works, because you get very high concentrations. What they don't do is spread out from the implant to cover the entire brain. And instead, when you put an implant in the brain like this, and this is a special measurement which I won't describe completely, but here's an implant that we put in the brain, and then we can track where the drug molecules go. And so the drug molecules are released, and you get very high concentrations of drug right around the implant, very high concentrations, higher than anyone's ever measured in the brain. And that's why they're effective. But they don't go very far. If you look only a millimeter or two, a very small distance away from the implant, you find no drug molecules. All of the drug molecules stay inside. And those of you who have good intuition in physics will understand this. What we're pretty sure now happens is that drug molecules are released in the brain and they begin to diffuse, they begin to move away. But remember, these are chemotherapy drugs. They're highly reactive. They're killing cells, or they're being degraded by our normal metabolism. And so by the time the molecule has moved from here to here, it's all used up, right? And none of it can get this far. So we do a very good job of treating cancer when it's very close to the implant, but when there's a tumor cell that's this far away, it doesn't even know that the drug is there, right? And so these cells can start growing again and form a new tumor. So how are we going to get this drug molecule, these drug molecules, which are very effective at the place where you've put them, how are we going to get them to go through the brain? Well, this is one way to do it, and I like this idea, and you can tell me if you think it's going to work. But instead of making a big wafer the size of a dime, what if we shrunk it down? So instead of making a big wafer, we made tiny, tiny particles. And that's what these are. These are tiny, tiny particles that have all the properties that I described for you before. They're made of polymers. They dissolve slowly in water. They contain a lot of drug, but they're very, very small. In fact, this scale bar is 500 nanometers, or a half of a micron. A micron is how big a bacterium is. You can't see it. You can't see bacteria, right? They're small. These are much, much smaller than that. In fact, they're the size of a virus that causes a cold. They're the same size, but they're totally synthetic. They're made of polymers. They're packed with drugs. And if you put them into the body, they'll release the drug in the same way I showed you before, and then they'll slowly dissolve. And the way that we imagine getting these into the brain is by infusing them. So here's the treatment that we currently have, where a surgeon implants the polymer and you get a zone of drug. The drug is shown as red here, right around where the polymer is. So these are very, very good at treating tumors that are right there by the implant. But if we could break this down into 1 billion pieces and then infuse them in the brain in a way that each piece is able to penetrate out into the brain, the surgeon could, instead of implanting this device, he could put a needle or a catheter into the brain. He could infuse our tiny particles. The tiny particles would crawl out into all reaches of the brain. And once they were there, they would sit in these locations and release their drugs in the same slow way I described before. And they would kill all the tumor cells that are in the vicinity of this small particle. Have the same advantage in not causing side effects, but a big advantage in terms of penetrating. And so we're very optimistic about this new technique. When we've tested it in animals, Andy Sawyer and his wife Jenny did these experiments and showed that they could get much better treatment of brain tumors in animals using this new approach where we make tiny, tiny particles and infuse them all through the brain. In fact, 30% of the animals are completely cured of brain cancer in this particular experiment. And we're working with physicians at the Yale New Haven Hospital to try to bring this therapy to patients hopefully within the next few years. Now, that's good for brain tumors, but how would you treat tumors in other places? You need to make these tiny particles smart somehow, right? In that example I just showed you, the particles didn't need to be very smart because the surgeon was smart. He knew where to put them into the brain, right? But what if you could make a particle that was as smart as a surgeon, so that you inject it into the body, you inject it into the blood, it goes all over the body, and it finds the tumor, right? That's what we're working on next, and we have some particles that appear to work that way. These particles are red, and this is a mouse, and we injected it into the tail vein of a mouse, so it circulates all through the body. And this is a mouse that unfortunately has a tumor. You can't see it yet, but now you see the tumor right there. Where's all the red particles? Mainly in the tumor, right? So you can make these particles that you can inject into the body. They're very small, they circulate very easily, and they're smart enough to know where the tumor is, and when they find the tumor, they stay there. And we're hoping that this could be a new way to treat cancers that appear any place in the body. This is work from a cancer doctor named Chris Holmes and Yang Deng, who designed and built these smart particles and are testing them now for use in treating prostate and ovarian cancer. I'm going to change directions a little bit and talk. This is a new way that medicines are going to be delivered. It's maybe not so new because some of these things I've described are now being used to treat cancer all over the world. But I think these ideas are going to become more widespread in other areas of treating diseases as well. And that's why I call it medicines of the future. This next part is a little bit more futuristic. And it involves not just using drugs, but using naturally occurring molecules that our body makes in order to build itself, really. So what I listed here are two proteins that are growth factors. Vascular endothelial growth factor. This is a molecule that's made naturally in your body all the time, and one of its jobs in your body is to promote the growth of new blood vessels in regions of your body that's not getting enough oxygen. This molecule gets released, and new blood vessels grow. So where might be a place in your body where you need new blood vessels? In adults, mainly we have blood vessels everywhere. We don't need new ones all the time, but there's some diseases you get where you need new blood vessels to be recreated. You think about what diseases that might be. Yeah. In the heart, right? Leading cause of death in the United States is heart disease, and most of heart disease is because there are blood vessels in your heart that stop working right, and they're not carrying oxygen to the heart tissue in the right way. But if you could deliver vascular growth factor there, you might encourage the heart to grow new blood vessels. Right? That's exactly right. So that's the example I want to talk about. Nerve growth factor is another kind of molecule that your body makes naturally, and what it does is promote the survival and maturation of certain kinds of neurons. So your body, particularly in your brain, is making nerve growth factor all the time, and this molecule helps your neurons to live healthy lives and to stay in your brain and keep doing what they're supposed to do. In some cases, though, that doesn't happen. And so you'd like to be able to promote survival. And I talked about one example of that in Parkinson's disease. Another example where cells are not surviving and they should is Alzheimer's disease. Right? Cells in your brain are slowly dying, and you're losing function because of that. So you already know how to make delivery systems for proteins, because Jill showed you how to do it on the bench outside. This is that same experiment or demonstration that she showed you. These are tiny particles that you make by a very simple method of taking alginate, a polymer that's a naturally occurring polymer, a polysaccharide, and dropping it into a bath of calcium. And the calcium causes the polymers to all link together, and so they form tiny particles. And you can see those particles because Jill showed you how to make them with food coloring in them so that they appear in different colors. But you can also make them with growth factors like vascular endothelial growth factor, this molecule in them. And it turns out this is a better way to make factors like this because these proteins are very sensitive. If you made them using the kinds of approaches that Senya showed you, where you synthesize polymers in organic solvents, these things would not survive. These drugs are much more sensitive, so you need gentle methods like this one in order to make particles. But you could use these particles in the same way that I described before. In fact, if you take a bunch of these particles and you inject it into the skin, they release the molecule and they make new blood vessels form in the skin. And here you can see a new blood vessel that we produced in a mouse just by injecting a few of those particles that contain vascular endothelial growth factor into the mouse. And here we could measure how many, and we've designed systems that cause a lot of blood vessels to grow locally. So what would you do with this? Well, what if instead of doing surgery, bypass surgery, to add new blood vessels to the surface of the heart, when you found out that a heart was starting to lose its blood vessels or become unhealthy, you could just inject some of these particles right on the surface of the heart and cause new blood vessels to form, delivering more oxygen to the tissue. So we think that this might someday, if we keep working on it, be a valuable approach for treating certain kinds of diseases. I mentioned, I'm going to give one last example, and then I'll hopefully leave time for some questions. But I mentioned Parkinson's disease, and I mentioned ways of treating Parkinson's disease at the very beginning with a deep brain stimulator. And that's been a tremendous advance for patients, but it doesn't treat the underlying disease. What's really happening in the brain of patients that have Parkinson's disease is that the neurons that are responsible for helping you control your motor system are starting to die. And those neurons are shown in this picture of the brain. In a normal brain, you can see lots of those neurons. They're the brown cells here. And in someone that has Parkinson's disease, you find many fewer of them. And it's the loss of those neurons in your brain that's causing the disease. The brain, these cells have a pigment, which is what makes them brown. And so if you look, this is really in the brain. You can see here's the region where those cells are, and those cells are disappearing. Right? So what if you could encourage, one, discourage those cells from dying, and two, put new cells back in this area of the brain where it's depleted? One way to do that is by using these kinds of systems I described before, but using it in a different way. And I'm going to show you the end result. But this is a cluster of brain cells that we've manipulated outside the body. So this is many thousands of brain cells, the kinds of brain cells that are lost in Parkinson's disease, that we've maintained outside the body in a way that they form a little cluster of tissue. And what you can't see here is that this little cluster of tissue is all held together with tiny polymer microparticles like the ones I've been describing before. So if you could look inside this, the microparticles are green, the cells are red, for example. You can see these little particles inside. And these particles are not only holding the brain cells together, they're also releasing this molecule called nerve growth factor, which helps cells survive in the brain and keeps them in their mature state. So here we've made a little piece of tissue which we think might resemble some aspects of the tissue that's lost in Parkinson's disease. We've made it outside the body using exactly these same kinds of methods I told you before are used for making drug delivery systems. Now, if you put this back into the body, what happens is the nerve growth factor comes out of the tiny particles, it accumulates in the brain, it comes to very high concentration, and it helps those cells survive in the brain for a much longer time than they would if we just put the cells in on their own. Now, this is a much more in the distant future kind of treatment. But we think someday it might be possible to combine these kinds of materials I talked about before with cells that are transplanted just like our brain tumor implants are implanted in the brain, but these are transplanted back into regions of the brain that need new cells and could help people recover from diseases like Parkinson's disease and Alzheimer's disease. The question you would ask is, where do the cells come from? And that's a challenge for the future. But there are places around the world, like Yale, where biologists are working in centers like Yale's Stem Cell Center to try to learn how to produce these kinds of cells in ways that can be to produce cells like these brain cells that can be transplanted back safely into people. So we think we're ahead of the curve in terms of engineering here, and that we have to wait for the biology of identifying the right stem cells to catch up with us. Just one last slide to show you some people. I've mentioned people as we've come along, and you met Jill and Senya, who are both doing research in this field. And I just show you some pictures of Yale undergraduates. And we've had high school students who've come into the laboratory to work on these projects with us. And one of the great pleasures for me is not only being a scientist, but being able to work with very smart, very talented, very highly motivated people to try to make these kinds of visions for the future a reality. Thanks for your attention. I think I've left a little bit of time for questions if you have any.